This is the usual attendance. Okay, shall we start? So today you will be having a session on microbial virulence and pathogenesis. So in year 1 you have studied the basics of microbes, right? I think it's better for you all to go back home and just read those slides, okay? Especially bacteria, virus and fungi, concentrating more on bacterial parts, okay? Whatever has been dealt in year 1. So that will make you much easier to understand these 2 weeks problem, okay? So here basically the slides will be concentrating on few definitions, okay, which is important in case of infections. Morphology of a bacteria, which is a revision from year 1. Study of bacteria, how do you study bacteria? Especially we will look at some stains that are being used. Then virulence factors. So you know that there are some bacteria which are normal microflora, right? There are some groups that are pathogens. So what makes them pathogen? Okay, so that explains the virulence factors. It's also called as virulence determinants. Then the determinants of bacterial pathogenesis. So what is the cycle involved from the time the host is exposed to the agent, that is the infectious agent. Once it enters inside, what are the steps that are involved? And finally, a short flavor on virulence of virus and fungi. Okay, coming to definitions, and I think by now you all know what are these terms. Pathogen means an organism which is capable of causing disease. Pathogenesis, it is the mechanism involved in disease. Okay. Pathogenicity is the ability of the organism to cause disease. Okay. Coming to virulence, virulence is the quantitative ability of the organism to cause disease. Okay. So you see some pathogens, they are less virulent. Some are highly virulent. Okay. Toxigenicity, so from the name itself, ability of an organism to produce toxin. Incubation period, I think all of you would have discussed in your problem, right? Incubation period, it is the time period which starts from the time the person is exposed till he develops the symptoms. So that is the incubation period, which varies from one microbe to another. And carrier is a person who harbors that, who has the pathogen, but he does not exhibit any symptoms. So that person is called as a carrier. So coming to morphology, so this is a revision. So what are the different shapes of bacteria? So if they are round in shape, they are called as coccus. Okay, so more than one is cocci. If they are arranged in two, they are called as diplococci. And there is something that bacteria called as streptococci, which is arranged in chains. Another morphology is bacilli, that means they are rod shaped. And we have spirillum, which are spiral shaped. Okay, so how do we study these bacteria? So there are different types of stains that are used to study the bacteria. So these stains, they are again categorized as simple stain, negative stain, differential stain and special stain. And there is something called as wet preparation. Wet preparation means here we don't use any stains. Okay, we take the bacteria, we put some saline and we see under the microscope. So this wet preparation is basically done to look if the bacteria or the microbe is moving or not. Okay, or to look for any ova or cyst that you might have studied in parasites, okay, the stages of parasites, or to look for molds, which is a type of fungi. So this is the wet preparation. As I told you, the bacterial inoculum, on this we place this cover slip and see under the microscope. So that is wet preparation, or it is also called as wet mount. And these are the stained preparations where we use different types of stains. Okay. So here basically it is showing you the procedure. You take the sample, you put it on a glass slide, cover it with the cover slip and look under the microscope. So this, I think all of you know, this is a glass slide, okay, and these small structures you see, they are very thin, they are called as cover slips, okay, and these are some stages, these are the cysts that you see in case of protozoa, okay. And there is something called as hanging drop slide. So hanging drop slide is this basically used to look for the motility. So here you see that the slide has got a slight depression, it is like a concave in the center, okay. Here you see that it is fully straight, right, but here there is a slight depression. So what do we do is we just take an inoculum from this and we place it on the cover slip and we put it over that concave structure, okay, and then we see under the microscope. Coming to stain preparations. So coming in to stain preparations, there is something called simple and differential. So simple means we are using just one stain, okay, so that's simple. So all the bacteria will take up only one color. So if you are putting crystal violet, all bacteria irrespective of cocci, bacilli, it will take all blue color, okay. So that's simple stain. So here you can see all the bacteria have taken blue color. So this will tell us the morphology, the arrangement of the bacteria. The second is differential stain. So from the name itself, it is used for differentiating, okay. So here you can see this picture, you can see two different types of bacteria, right. One is red in color, the other one is purple or dark blue in color, okay. So an example of this differential stain is gram staining method. I think you have come across gram stain, right. So based on the gram staining method, you can look at the size, shape and arrangement of the bacteria. So what is the principle of gram stain? Again revision from year 1. It is basically based on the peptidoglycan, the peptidoglycan component present in the cell wall. Okay. So if the peptidoglycan content is huge, okay, then what happens? Here you can see that in gram stain we have 4 steps: crystal violet, iodine, decolorization and counter stain, okay. So first when you add crystal violet, both the bacteria will take up crystal violet stain, okay. Then iodine is added to, it acts here as a mordant. So it will help the crystal violet to get attached to the cell wall, okay. And then coming to the third step, decolorization. So here when you add an acid, what happens? The thin layer, the crystal violet from this thin layer gets washed out, okay. Whereas from the thick layer remains as it is, okay. And the final step is to counter stain with safranin. So when you counter stain this with safranin, the one dye which has been washed out, this bacteria will take up safranin and it will look pink or dark red in color, okay. And the one with thick peptidoglycan, it will retain the primary stain, that is crystal violet. Hope you all understood this, okay. So we will be demonstrating this in the lab, okay. So basically the principle is on peptidoglycan content, the composition present in the cell wall, okay. So why we have to do this gram stain? What is the importance of gram stain? First and foremost, it helps the clinician to choose the antibiotic. So for gram positive bacteria, it's different set of antibiotics. For gram negative, it's a different set, okay. So here once we do the staining procedure, it hardly takes 2 to 3 minutes, okay. So you know whether it is gram-positive bacteria present or gram-negative. So accordingly, the clinician can start up with the antibiotics. Whereas culture, it will take time. Okay. So this helps in the choice of antibiotic. Second, it helps in the selection of culture media. So you can see on the right side, there are different types of culture medias. So this media are used for the growth of bacteria. So once you get a sample from the hospital, we inoculate these samples on these different culture media. So how do we select this culture media? You can see different colors, right? So how do you know I have to select this or this or this? That again is based on the gram stain. So if it is gram-positive bacteria in clusters, and if we are suspecting, yes, it is gram-positive cocci in clusters, then it could be Staphylococcus. So based on Staphylococcus, we select particular media. Okay. So it helps in selection of culture media, and it also helps in the judgment of specimen quality. Okay. So this you will study in unit 2. When a person comes or when a patient comes with respiratory infection, right? He coughs, he has sputum. So we ask the patient to provide sputum. So the patient provides in a small bottle sputum. Okay. And when we do a gram stain, if we see that the squamous epithelial cells are a lot, that means it is not sputum, it is something from the oral cavity. So then we can tell the patient, yes, this is not fit for culture, right? You have to provide another specimen. Okay. So thus gram stain can help in judging the specimen quality. Next is negative stain. So from the name itself, negative, that means the bacteria does not take up any stain, it is only the background that takes up stain. In simple stain and differential stain, you saw that the bacteria takes up the stain, right? But here, from the name itself, negative, the bacteria is negative, no stain for the bacteria, but the background takes up the stain. Next is the special stain. So if we want to look at flagella or if we want to look at the capsule, we use special stains for that. So those are called as special stains, capsule stain, or spore, endospores, exospores, you have studied in year 1, right? So for spores, to look at the spores, we do special stains. Okay. Coming to the structure of bacteria. So by now you know the structure of bacteria, right? So in the structure of bacteria, the most important is cell wall, right? So cell wall, based on peptidoglycan, we do a gram stain, and the bacteria are divided into gram positive and gram negative. So the cell wall is called as gram positive cell wall and gram negative cell wall. So when I say gram positive, so one of the example is Staphylococcus aureus. So from now on, you have to know the names of bacteria. Okay. So for gram positive bacteria, it is Staphylococcus aureus. For gram negative bacteria, Escherichia coli, Shigella and Salmonella, which you have already come across in your present problem. Okay. So they both are gram negative bacteria. So what is this gram positive cell wall? What is the composition of gram positive cell wall? So this gram positive cell wall, you can see here, this is the cell wall. It is made up of peptidoglycan, which is a thick layer, followed by lipoteichoic acid and teichoic acid. So what is the function of peptidoglycan? It provides support to the cell, bacterial cell, okay? And teichoic acid, it helps in the attachment of this bacteria to the host cell, and it is also responsible for shock, okay? And the third component is lipoteichoic acid. So from the name itself, lipo means lipid. So if the lipid is attached to the teichoic acid, it is called as lipoteichoic acid, and its function is, again, it is antigenic, cytotoxic and adhesive. Okay, so it helps in the attachment of the bacteria to the host cell, and it is toxic to the cells as well as antigenic. So, coming to gram-negative bacteria. So, in gram-negative bacteria, so you can see here, this is gram-negative bacteria. It has got a thin layer of peptidoglycan, then it has got the lipoproteins and outer membrane layer. Okay. So, this is the composition of gram-negative cell wall. Okay. So, again peptidoglycan in gram-negative cell wall, it is very thin. Okay. It comprises only 10%, whereas in gram-positive cell wall, it comprises around 90% of the cell wall. Okay. Lipoprotein layer, so you can see here, this is lipoprotein. Okay. So, what does it do? Its function is to stabilize the outer membrane. Okay. So, you can see it is attached to the periplasmic layer as well as the outer membrane. So, it helps in stabilization. And outer membrane. So, what is this outer membrane? It is the outermost layer. Okay. And this consists of outer membrane proteins and lipopolysaccharides. So, most important is lipopolysaccharides. What are these lipopolysaccharides? So, lipopolysaccharides are also called as endotoxin. Okay. So, this is specific to gram-negative bacteria because it is a component of cell wall. Okay, it is a component of gram-negative cell wall. So, this lipopolysaccharide, it has got three parts. Lipid A, which is inside, then comes the polysaccharide layer and O antigen, which is outside. Okay. So, here comes the O antigen and here comes the lipid A. Okay. And the most toxic part of this is lipid A. Okay. So, lipid A has got the toxic property in case of gram-negative bacteria. So, when you compare gram-positive and gram-negative cell wall, so in gram-positive cell wall you have thick peptidoglycan, teichoic acid, lipoteichoic acid. Okay. In gram-negative cell wall, the peptidoglycan is very thin. Okay. So, along with thin peptidoglycan, it has got outer membrane, right, outer membrane as well as lipopolysaccharide, which is a part of outer membrane. Now, so coming to the structure and function, I think most of you know this, right, because this is a continuation of year 1. Okay. So, peptidoglycan, which gives support, outer membrane, especially the endotoxin, which is a toxic component. Then there is a capsule which protects the bacteria against phagocytosis. Pili, okay, there are two types. One is it helps in the attachment of the bacteria to the host cell. Second, it plays a role in conjugation. Do you remember conjugation? What is conjugation? Yes, okay, right. So, I will show you the slide. Okay. So, we have already studied how the genetic information is transferred from one bacteria to another. In that, one of the mechanism is conjugation. Next is flagella or flagellum. If it is one, it is called as flagellum, which helps in motility. And the last one is plasmid. So, which genetic mechanism makes use of plasmid? Year 1? Transmission, transduction. Okay, let us see. Okay, fine. So, this is just a small question. If you can tell me which one of the following is the most accurate description for gram positive and gram negative bacteria. I think it's the most easiest one. A, okay? The first option itself, you don't have to take a picture, okay? It's the most easiest. Anybody, I think, in the class can answer this, right? So it is gram positive has got a thick peptidoglycan and gram negative has got a thin peptidoglycan. Okay, coming to virulence. So why are we studying about this virulence? What is the importance of virulence? Okay. So virulence factors, they help the bacteria to invade the host, to cause disease and to escape from our immune system. Okay. So they have all these properties, the virulence factors, they exhibit all these properties. So what are these virulence factors or virulence determinants? Indirectly, they are the structures, okay? They are the structure of bacteria. So we have adhesins, capsule, two different types of toxins, flagella and enzymes. So these are few, there are so many, depends upon one bacteria to another, okay? So when it comes to adhesins, adhesin, I think all of you understand the term, right? Attachment. So what are these adhesins? There are two types. One is fimbrial and non-fimbrial. So fimbrial, you can see here this hair-like structure with a protein. So this is fimbrial adhesin. And the second one is just an adhesin, okay? So this is the protein which is present on the cell wall of the bacteria, which helps to get attached to the host cell. Next is pili, okay. So these are the hair-like structures, and as I mentioned, it helps in the attachment as well as in conjugation, okay. The genetic material can be transferred, or genetic information can be transferred from one bacteria to another by this pili, which is also called as sex pilus. Coming to capsule. So we all know what is the function of capsule? Antiphagocytic, right? So it prevents it from bacterial ingestion, okay? So the second role, it helps in the adhesion. Third, it is sometimes used in the preparation of vaccines. So when you study further in your vaccines, you will come to know that there are some organisms whose capsule is taken and they use it for preparation of vaccines and for specific identification. So I told you in special stain, if you want to look at capsules, we use capsular stain, okay, and we look at the capsule. Then coming to flagella. So what is flagella? It is a long hair-like structure, and the function of flagella is to move, right? So by movement, what the bacteria can do? It can get attached or adhere to the host cell. It helps in the invasion of the host cell as well as in chemotaxis. Okay? So these are the functions of flagella. Now coming to toxins. So bacteria, they produce different types of toxins. One you have already studied, that is endotoxin. Okay? And this endotoxin is made up of lipopolysaccharide, and this is present in all gram negative cell wall. Okay? Remember that. Whereas exotoxins, from the name itself, exo means released out, secreted outside. Okay? So exotoxins, they are again enzymes that are excreted into the environment. So these exotoxins can be produced by gram positive bacteria or gram negative bacteria. Okay? So both the bacteria can produce exotoxin. So coming to endotoxin in detail, as I mentioned, they are lipopolysaccharides present in gram negative, and the most toxic component in that lipopolysaccharide is lipid A. Okay? And you can see here, okay. So whenever there is cell lysis or when this bacteria breaks down, these endotoxins are released. Okay, so they are part of bacteria and they are heat stable. So what are the functions of these endotoxins? Three important functions. They can activate macrophages, activate complement, and they can activate tissue factor. So what happens if they are activated? We might have fever and hypotension, because here on activation of macrophages, these macrophages will release interleukin 1, TNF, nitric oxide, leading to fever and hypotension. There will be activation of complement. I think in year 1 you have studied complement, different types. Complement system? No? Yes, okay. So here, that's why I told you that you just go and revise, okay? So you can remember, it will refresh your knowledge, okay? So activating the complements can again lead to hypertension, edema. There will be chemotaxis, and it can also activate the coagulation cascade, leading to disseminated intravascular coagulation. Okay? So these are the functions of endotoxins, ultimately leading to shock. Okay? So in gram-negative bacteria, the most important component that can cause shock is endotoxin. Coming to exotoxin. So what are these exotoxins? They are proteins, okay, and they are heat labile. You understand what is heat labile and heat stable? Huh? Okay, so stable means it is not affected by heat. It is stable, okay, whatever is the temperature. Whereas labile means it is affected by heat, okay. So exotoxins are heat labile and they can be converted into toxoids, whereas endotoxins, no, okay. So what are these toxoids? It is an inactivated toxin, okay. So that's called as toxoid. You might have heard for tetanus we use TT. What is full form of TT? Tetanus toxoid, okay. So what are the modes of action of these exotoxins? They can cause spores in the cells, host cells, inhibit protein synthesis, and they can also have effects on nerve muscles, okay. So these exotoxins, depending upon their site of action, they can be mentioned as cytotoxins. If they are working on nerves, they are told as neurotoxins. If they are working on the GI tract, they are called as enterotoxins, okay. So depending upon the system, their names change, but overall they are exotoxins, okay. So this is an example of exotoxin, that is botulinum toxin, which is produced by a bacteria called Clostridium botulinum. In your previous problems, you have studied how a muscle contracts and relaxes, right? So what is the neurotransmitter that is required for contraction? Acetylcholine. So what does this botulinum toxin do? It will inhibit the release of acetylcholine. So when the acetylcholine is not released, what happens to the muscle? It's always relaxed, right? So you can see here the baby with hypotonia, okay. So that's also called as flaccid paralysis. So since this exotoxin is working on the nerves, it is a neurotoxin, okay, produced by this bacteria. So from the name of the bacteria, they have given this botulinum toxin the name, okay. So these are the differences between exo and endotoxin, which you can go through. It is the same what I had explained. Coming to enzymes. So what are these enzymes? These enzymes, how do they help us? They help in spread of pathogen, okay, to invade. So, one of the example which I have mentioned here is collagenase. So, what this collagenase enzyme will do? It will destroy the collagen. So, it is very easy for the bacteria to spread. Okay, this is refreshing your mind. Okay. So, the three different mechanisms of gene transfer. Okay. So, one is conjugation. For this we require sex pillars. Okay. So, you can see a plasmid, part of it enters into another. Okay. So, this is conjugation. For transduction, it is bacteriophage. Okay. Bacteriophage, which has any genetic information for any resistant genes, toxins or virulence factors, can transmit the information to another bacteria. So, that is transduction. And the third one is transformation. Okay. So, from the bacteria which is dead, the genetic information is transferred to a competent bacterium. So, these are the three different mechanisms of gene transfer. So, now you know the structure of bacteria, the virulence factors of bacteria, right. Now, coming to pathogenesis. So, what is the cycle? First and foremost, what do we require? We require host. Second, pathogen. And third, a suitable environment. So, when these three things are present, then of course, there can be a infection. Okay. So, first and foremost, exposure of the host to the microorganism. So, once the susceptible host is exposed to a microorganism, this organism enters into the human body, right. So, that is different modes of transmission. Maybe you would have discussed different modes of transmission of microbes. So, once it enters into the human body, what is the next step? It has to get hold of host cells, right. So, that is adhesion. Okay. So, after it gets hold, what is the next step? It has to invade. It tries to invade. It tries to cause some inflammation. Okay. And there are some bacteria that can stay within the cells or within the macrophages. Okay. So, that is called as intracellular survival, and it can produce toxins, and in some diseases you will have immunopathogenesis. So, I will tell you what is that. So, coming to transmission, why should we know transmission? I think all of us have faced pandemic, right. So, we knew that. How is it transmitted? How was it transmitted? Yeah, that is basically through inhalation. So, what were we advised to do? Wear masks. Okay. So, indirectly how does it help us to prevent or interrupt the chain of transmission? Okay. And what are the different modes of transmission? Human to human. Okay. That could be through direct contact, blood, or from mother to the baby, or non-human to human. It could be from vectors, fomites. Okay. And what are the different portals of entry? Could be respiratory, gastrointestinal, through abraded skin. Okay. The normal skin is always protective, but if there is any cuts or wounds in the skin, so that could be a route for entry of microbes, and through genital tract. Okay. So, once it is transmitted, second is adhesion. So, for adhesion you have already studied there are adhesins. Okay. There are fimbrial adhesins or there are other adhesins, okay, depending upon the type of bacteria. Then invasion. So, invasion is caused by what plays a role in invasion? It is enzymes. Okay. Okay. So, coming to some bacteria, even though they are engulfed by macrophages, they stay happily within the macrophages. Why is that? There are some mechanisms that happens within the macrophages. There are some bacteria, for example, you have heard Mycobacterium tuberculosis, okay, which causes tuberculosis, right. So, they have some signals, or they produce some signals which makes them escape to the cytosol of the macrophage, or they prevent this lysosome endosome attachment. Okay, so like that, when these normal phagocytic mechanisms are prevented, the bacterium can survive within the cell. So coming to toxins, I have already mentioned endotoxins, exotoxins, immunopathogenesis. So what do you mean by immunopathogenesis? So here the symptoms of the disease is not caused by the organism, okay, but how? It is due to the immune response to the presence of organism. Okay, so that's called as immunopathogenesis. An example of that is rheumatic fever, which you will study in detail in your further units. Then there is something called as biofilm. So what are these biofilms? They are aggregate of interactive bacteria. So the bacteria, once it enters into the human body, they come close together, they develop, and there is a film-like production. And what is the importance of this bacteria? It protects the bacteria, biofilm protects the bacteria from antibiotics. So it's very difficult to treat such bacteria that produce biofilm, okay? And it will also protect the bacteria against host immune defenses. So this is just a short video just to make you understand what is this biofilm.
Infection. The bacteria may be exogenous and, for example, transmit from the hands of the healthcare professional. But they can also be endogenous, then the person's own bacteria from the rectum or the urethra, for example, contaminates the catheter. When enough bacteria have developed, they form a biofilm. This takes approximately 48 hours. Biofilm increases the risk for infection. In addition, microbes in biofilm are more resistant to antibiotics and the patient's own immune system than microbes not forming biofilm.
Okay, so you understood this is how a biofilm is formed. So if a bacteria produces such a biofilm, then it's very difficult for the clinician to treat that particular organism. Okay. So now we have studied everything about bacteria, right? So an example of a bacteria which is mentioned in your problem is Shigella, okay? So this Shigella, what does it cause? It causes something called as shigellosis or bacillary dysentery. And the second point that you note here is, for this bacteria to cause infection, just 10 to 100 organism is sufficient. There are some bacteria which require 1000 or 10,000. Okay, so this is called as infectious dose. Okay, so for Shigella, just 10 bacteria is enough. So maybe in Amina's sandwich there was just 10 bacteria, okay, that developed this infection. And the transmission is feco-oral. Okay, when you eat contaminated food. And the virulence factors that are present in this bacteria is, because we know that Shigella is gram positive or gram negative? Gram negative bacteria. So one of the virulence factor that has to be present in this gram negative bacteria is endotoxin. Okay, so it has got endotoxin and it produces another toxin called as Shiga toxin. Okay, and it has also it has something called as intestinal adherence factor, which helps it to adhere to the host cell. Okay, and you saw that initially Amina was having watery stools, right? Later it progressed to bloody diarrhea, right? So what would be the reason? The bacteria try to invade, okay? When it tries to invade, it can cause bloody diarrhea. So that is the critical factor in pathogenesis of Shigella infection. Okay. So when Amina had all these signs and symptoms, the most important sample that is requested by the clinician is stool sample, right? So when we receive the stool sample from the patient, the first and foremost thing that we do is stool microscopy or stool routine, okay? So in stool routine, first we see how is the stool, whether it is watery, whether there is blood or something like that, macroscopically through the naked eye. Then microscopically we look at anything else is present, WBCs are present or RBCs are present, okay. So if it is due to Shigella infection, WBCs and RBCs will be present, okay. So then we would like to know what type of pathogen is present. So for that we do stool culture. We use a specific media called McConkey's agar, which you will study in your lab, okay. So when you inoculate the organism, now here you can see this is McConkey's agar, a pink culture medium, okay. When you inoculate the stool sample on McConkey's agar, you will find that they are non-lactose fermenting colonies. So you will see some colorless colonies if it is Shigella, okay. And on gram stain, they are of course gram negative bacilli, okay. So based on this results, as well as we do some biochemical test and we do an agglutination test, based on that we say that yes, this is Shigella. There are different species of Shigella, okay. Okay, coming to this slide. 35-year-old male presented, no need to take a picture, this is very easy, 3-day history of fever, chills, abdominal pain and bloody diarrhea. It's something which you have studied in this present problem, right? So there is a history that this patient consumed unpasteurized dairy product, okay, and the stool culture is positive for Salmonella. So what is Salmonella? Gram negative, okay. So describe the gram stain finding of this organism. It is a gram negative bacilli, okay. So now you know two types of bacilli, which is Shigella and Salmonella. They both are gram negative. So next, name 2 virulence determinants present in this bacteria. Endotoxins, very good. Another one? What do you think? You have not studied Salmonella, but still attempt. You can just rephrase, yes?
Enzymes for invasion.
Enzymes for invasion, yes. Anything else? Sorry? Yes? But what? What is the virulence? We have to be specific with the names, right? So one is endotoxin, the other one there could be enzymes. So once this bacteria enters, what happens? What is required for adhesion? Adhesins, right? Any bacteria enters into the body, it requires adhesins. So which you can come up with is, you know, it is gram negative bacilli, so endotoxin could be a virulence factor, or there could be, because it is bloody diarrhea, it is causing invasion, so there could be enzymes, or once it enters in, it has to adhere, so there could be adhesins, okay. This is how you have to think, okay. So coming to viruses. So now you know what are viruses, right? I will not go through this, you know the structure, you know the basis of classification, right? So even viruses, they have some virulence determinants, okay. Some viruses, they cause infection. So they have surface proteins. What do these surface proteins do? Adhere, attach to the host cell. Then How do they enter in? It is by endocytosis. You have seen virus replication mechanisms, right? You have studied, right? Endocytosis. Then these viruses, they can evade immune response. That is, they can inhibit presentation of their antigens. So the host immune response will not recognize it. Okay? And what they can do, they can manipulate the host cell. They can manipulate the human host cells and they can take control over that. Okay? And immunomodulatory proteins. So they can produce these immunomodulatory proteins which can inhibit their signaling, okay, which can inhibit antiviral signaling pathways. So that is with respect to viruses. Coming to fungi. So in fungi you have studied different types of fungi, right? How the fungi are classified? Yeast, molds, huh? Pseudo, okay. But the third, purposely I didn't put the third part. Something which grows in both the temperature?
Dimorphic.
What? Dimorphic. Okay. So we have yeast, we have mold, and the third group which is dimorphic fungi. So what are the virulence factors which may be present in these? So one is glycoproteins which help in adhesion. They might have capsules which are again antiphagocytic, enzymes which help in invasion, and toxins which are cytotoxic. It causes necrosis of the tissue. Okay? So these are just a small flavor. Fungi, viruses, they have many more virulence factors, okay, depending upon the type of microbe. Hope all of you understood it. It's a bit exhaustive, but try to read year 1 and then go to these slides. So it will be easy for you to understand and adapt. Any questions? Anything you want me to repeat?
So virulence is the severity of the bacteria, like the effect of that bacteria on the host.
Yes.
It's
Okay.
not the quantity, it's the severity.
See, it is a quantitative ability. So one bacteria can be having only flagella. The other bacteria has flagella and exotoxins. So which one would be
based on
more
the
virulence
virulence,
factors? Yeah. So that's why it's called as quantitative ability. Any other questions? Yeah. Okay. So when we look at blood agar as well as McConkey agar is a differential medium. Okay. So this can, in stool we know that there is normal microflora as well as now there is a pathogen also, right. So based on the history of the patient, we usually choose McConkey's agar. So McConkey's agar can differentiate. So if it is Shigella, it can produce non-lactose fermenters. E. coli is a normal flora of human being, right? So E. coli will be a lactose fermenter. So if you see everything is lactose fermenter, that means it is not, it is a normal pathogen, okay? Now, normal flora. So when you come to the lab, you will see all these, okay? The different types of medium that is used, what is the use of different mediums, okay? And how do these bacteria grow, how the yeast grow, okay? And ultimately, yes, we came to know that this is Shigella, okay? So what is the purpose of knowing this, that it is Shigella in the patient? Huh? Yes, so ultimately it ends up with antibiotics, okay? So in the lab, you will study how the antibiotic susceptibility tests are done for such pathogens. Okay? Any more questions? Okay. So complements, these are the proteins, and you will be having a specific resource sessions by Professor Reda, the immunologist, and he will detail you about antigens and complements and further systems in the immune host defense. Okay?
So they're part of the immune system. I have another question.
Yes, dear.
which is,
Thank you. Others can leave.
Which is about the symptoms. So when a host comes in the body, The symptoms are by the immune system.
No, no, no.
They're by the host.
By the, yeah.
So-
No, no. Host is human.
Sorry, by the pathogen.
By the pathogen. So it can be by the pathogen itself, or there are
the
some diseases which it is due to immunity. Yes.
So what's the difference? Aren't they the same?
No. So when I say it is due to the pathogen, it is the pathogen virulence factors which is responsible for the signs.
Septic shock by bacteria.
Yes.
That's a by pathogen.
Yes, that is by pathogen. When I say rheumatic fever, you see that when this bacteria enters into the human body, so there is something called like immune mimicry. That means the antigens of this bacteria is similar to the cardiac tissue antigens. So the antibodies that are produced to kill this bacteria, which will also target the cardiac tissue.
So but the antigens are produced by the immune system.
No, no, antigens
By
are
the
part of
bacteria.
bacteria, yes, but they mimic something in the host cell.
Okay, so they're kind of trying to trick the body.
Yes, so what happens is the antibodies which are produced against this bacteria will also attack its own tissue. It is called as molecular mimicry, which you will study in your